Hello everyone. Today we will explore Hematocrit, also called packed cell volume or PCV. This simple test is central to diagnosing anemia and to understanding red cell indices. By the end of this video, you will be able to describe measurement methods, interpret normal and abnormal values, calculate indices, and classify anemia. We will emphasize clinical relevance so you can apply these concepts at the bedside or in the lab. Stay focused on key points and follow the step-by-step calculations and classification rules we present. Let's begin with a clear definition of hematocrit and a visual of centrifugation. Hematocrit, abbreviated HETCP or PCV, is the proportion of a blood sample occupied by red blood cells after centrifugation. When we spin blood at high speed, components separate, plasma on top, a thin buffy coat of white cells and platelets in the middle, and packed red cells at the bottom. The hematocrit is the fraction or percentage of the tube occupied by red cells relative to the total blood column. Microhematocrit methods use capillary tubes spun in a fixed angle or swing-out rotor and read with a hematocrit reader. Automated instruments can also report hematocrit, sometimes derived from mean cell volume and red cell count. A quick visual diagram will help. Plasma is pale, buffy coat is thin and white, and red cells form the dense dark column at the base. There are three common approaches: direct microhematocrit measurement, automated analyzer results, and estimation using simple rules. Direct microhematocrit measurement is fast and reliable when performed correctly with proper centrifugation and sealing. Automated hematology analyzers often report hematocrit directly or derive it from measured mean cell volume multiplied by the red cell count. A quick clinical rule of thumb is the rule of 3. Hemoglobin times 3 approximates hematocrit percentage in routine cases. Remember that leukemia, cold agglutins, or improper centrifugation can cause inaccurate hematocrits, so always consider pre-analytical errors. Next, we will define normal ranges and move into practical index calculations used to classify anemia. Key red cell indices are mean cell volume, or MCV, mean cell hemoglobin, or MCH, and mean cell hemoglobin concentration, or MCHC. Formulas are simple. MCV in femtoliters equals hematocrit percent times 10 divided by red cell count in millions per microliter. MCH in picograms equals hemoglobin in grams per deciliter times 10 divided by red cell count in millions per microliter. MCHC in grams per deciliter equals hemoglobin times 100 divided by hematocrit percent. Let's work a practical example. Suppose hemoglobin 10 grams per deciliter, hematocrit 30%, and red cell count 4 million per microliter. Apply the formulas. MCV equals 30 times 10 divided by 4 equals 75 femtoliters, which is Macrocytic. MCH equals 10 times 10 divided by 4 equals 25 picograms. MCHCHC equals 10 times 100 divided by 30 equals 33.3 grams per deciliter. Interpret indices to classify anemia. Use MCV thresholds. Microcytic less than 80 femtoliters. Normocytic 80 to 100 femtoliters. Macrocytic greater than 100 femtoliters. Assess MCHC to evaluate color. Hypochromic values typically fall below about 32 grams per deciliter, normochromic around 32 to 36. Combine indices with clinical data and severity. Hematocrit or hemoglobin thresholds guide severity, grading, and management decisions. Example classification. Our example with MCV 75 is microcytic and suggests iron deficiency, thalassemia trait, or chronic Disease mechanisms. Always correlate lab indices with history, exam, and other tests like ferritin, B12, folate, or hemoglobin electrophoresis where indicated. Summary: hematocrit measures packed red cell volume, can be measured directly or estimated. Indices are calculated with straightforward formulas, and together they help classify anemia for clinical decision-making. And that wraps up today's topic. If this explanation helped you understand the concept more clearly, make sure to like the video and subscribe to this channel for more simplified lab technology and radiology lessons powered by AI. We break down complex subjects in hematology, biochemistry, microbiology, pathology, X-ray, Connecticut, MRI, and diagnostic imaging, step by step, so you don't just memorize, you truly understand. If you have any questions or want a specific topic covered, drop it in the comments below. Until the next video, keep learning smart, keep thinking critically, and keep mastering diagnostics.